Thank you.
So you can.
Okay. So first of all, I would like to thank Mark and Annie for inviting me to give this talk today. So I would like to talk to you about Cereblon, the common target of multiple pharmacological actions of thalidomide and its derivatives. So here are the contents of my talk today. First, I will talk about the development of our affinity-based technology for drug target fishing, and then about identification of Cereblon as the key proteins in thalidomide teratogenicity using our bees technology. And then I will go on to the main issue, the mechanisms of actions of immunomodulatory drugs, called IMIDs for short, consisting of thalidomide and its derivatives. So here, I would like to briefly talk about the conventional method of protein purification from large protein libraries using liquid column chromatography. Multiple steps involving several types of column chromatography are required for conventional protein purification because the efficiency of concentration of each step is low, less than tenfold. So I will show you our example, our purification of NERF, just Annie told you. That is composed of 4 subunits using the 7 types of columns. So multiple steps take a lot of time and work, and also is very often cause the loss and the degradation of target proteins. So we wanted to a simple and more convenient method. So to achieve a single-step affinity purification of target proteins to overcome the problems of the conventional method, we developed novel bees matrices, which are now often called Handa bees. So we developed 2 types of submicron bees as novel matrices for single-step affinity chromatography. So first, we developed latex bees called SGBs, which are composed of styrene GMA copolymer at the core and poly-GMA at the surface. And then we developed magnetic bees called FGBs, which have magnetite nanocrystals in the core of the SGBs. So Handa bees have the advantage of being highly dispersive in solution and resistant to organic solvents. Large numbers, about 10 to the 7th of epoxy groups, are present on the bees and are used for ligand immobilization. So Handa bees are superior to conventional agarose bees in terms of their recovery rate, purification rate, and versatility. And compared to agarose bees, which are about 200 micrometer and have a polar structure, our bees are 1,000 times smaller and a non-polar structure. And our bees immobilize large number of chemical ligands and are highly dispersive and mobile in binding reactions. These characteristics generate the high recovery rate. And in addition, the surface property of the bees reduce non-specific protein binding, leading to high purification rate, as shown here. And moreover, the bees are physically and chemically stable, and the bees are able to immobilize chemical ligand in organic solvents and can isolate target proteins in buffered aqueous solution. These features enable single-step affinity purification of target from large protein libraries. The concentration rate is more than 1,000 times. So single-step affinity purification is performed by mixing the drug immobilized bees with cell lysate and then washing the bees several times through magnetic separation. And then the drug binding proteins including target molecule, are eluted by adding excess drugs or surfactant. A single 1.5 ml centrifuge tube is sufficient for the whole processes. This affinity-based technology has further advantages. First, high-yield recovery leads to rapid identification of drug-binding proteins by mass spectrometry. And second, binding specificity can be evaluated using the competitive inhibition assay and the competitive drug addition assays. I will introduce some practical examples of these assays later. And third, the binding modes and binding affinities can be roughly estimated by altering the binding, washing, and adhesion conditions. Based on the information obtained in this technology, we can often select a potential target which are involved in the drug actions from several drug-binding proteins. Well, here is a list of ligands, including pharmaceuticals and biomolecules and toxic chemicals for which we have identified the target using our BEAS technology and have published our findings as shown here. So now I would like to talk to you about the target of thalidomide, our identification of the target of thalidomide. Okay. Here, I will give you a little background of thalidomide. So thalidomide was developed and commercialized as a sedative in the late 1950s. And thalidomide causes the shortening or complete lack of the limbs and also causes the malformation of the ear. It's about 10,000 affected children were born worldwide. Thalidomide was later found to be an effective treatment for leprosy and multiple myeloma and is now approved for use in these patients. However, little was known about the mechanisms of actions of thalidomide regarding both its beneficial effect and side effect. So we attempted to identify the target of thalidomide, the target of thalidomide using our affinity BEAS technology. We immobilize a carboxylated derivative of thalidomide to our BEAS and mix with cell lysate. And after washing, the bound proteins were eluted by adding excess thalidomide, which is known as a competitive drug addition. We isolated two proteins that selectively interact with the thalidomide, which were competed by adding the excess thalidomide to the binding reaction, which is known as a competitive inhibition assay. So we then identify these proteins as DNA-specific, damage-specific DNA binding protein 1, called DDB1 for short, and Cereblon by mass spec analysis. And both proteins were confirmed by Western blotting using their specific antibodies. So the gene encoding Cereblon was originally identified to be associated with mild mental retardation, but little was known about its molecular function at that time. DDB1 was originally identified as a DNA repair factor and is now known to be a component of the CAR4-based E3 victin ligase complex. We tested the binding of thalidomide to recombinant proteins of Cereblon and DDB1 using our affinity BEAS technology. We found Cereblon directly bind to thalidomide, but DDB1 does not. And when the flag tagged Cereblon was expressed in cells and immunoprecipitated with using an anti-flag antibodies, endogenous DDB1 was co-immunoprecipitated with DDB1, Cereblon. So the result indicated that Cereblon forms a stable complex with DDB1. And next, we found that Cereblon forms the E3 victin ligase complex with DDB1, CAR4A, LOC1 using co-immunoprecipitation assays, as shown here. And the Cereblon bind to DDB1 in competition with DDB2. So DDB2, which is an established substrate receptor, and although I didn't include the data here, and Cereblon undergoes autoubiquitination. So these results strongly suggested that Cereblon works as a substrate receptor of the E3 victin ligase complex. And we also found that the ubiquitination of Cereblon is sensitive to thalidomide in a dose-dependent manner. So these results indicated that thalidomide bind to Cereblon and affect the enzymatic activity of the Cereblon E3 victin ligase. So here, I would like to talk about the interaction of Cereblon with thalidomide and DDB1. Thalidomide has optical isomers, S-thalidomide and R-thalidomide, and competitive drug addition assays, as shown earlier. So S-thalidomide has higher affinity for Cereblon than R-thalidomide, and that the thalidomide is bind to Cereblon via its glutamide moiety, but not via its phthalmide moiety. And affinity isolation and co-immunoprecipitation experiment indicated that the thalidomide bind to the C-terminal region of the Cereblon, and DDB1 interact with the central region of the Cereblon. So to test the effect of the, to examine the effect of the, and we investigated the involvement of Cereblon in thalidomide teratogenicity in vivo utilizing the zebrafish, because zebrafish has several advantages as an experimental animals. So rapid development, transparency of embryo, easy control of gene expression, possible to experiment on a large number of animals, employment for pharmacotoxicological studies. So the first things we did was to make sure that the Cereblon has a human, zebrafish has a human Cereblon homologue, and which can bind to thalidomide and interact with the human DDB1. So to examine the effect of thalidomide on zebrafish development, zebrafish embryos were grown in media containing thalidomide. So there were no gross morphological changes except for in the pectoral fin, as more clearly observed here by cartridge staining. And we also found that the development of the otic vesicle, zebrafish equivalent of the ear, is significantly affected. So these results indicated that thalidomide causes similar developmental defect in zebrafish as in humans. So we then knocked down of Cereblon by using the antisense morpholino oligonucleotide, or AMO, which inhibit Cereblon protein synthesis by selectively binding Cereblon mRNA. And we found that knockdown of Cereblon causes similar developmental defect in pectoral fins and aortic vesicles as thalidomide. And these defects were rescued by co-injection of AMO-resistant mRNA. And CAR4A also causes similar developmental defect. So these results suggested that the Cereblon E3 victim ligase complex is involved in early development and is a target of thalidomide teratogenicity. So far, the association between the silencing of Cereblon and thalidomide teratogenicity has been established. This indicates that the binding of thalidomide to Cereblon causes teratogenicity. So, however, the next question was, how can we really prove that the Cereblon is a true target of thalidomide teratogenicity? If teratogenicity is suppressed by the expression of a Cereblon mutant that does not bind to thalidomide, but is otherwise fully functional, we can conclude that the Cereblon is a bona fide target of thalidomide teratogenicity. So we were able to identify a Cereblon mutant that does not bind to thalidomide, but is otherwise fully functional. So we found that the tyrosine 384 and tryptophan 386 of Cereblon are important for its binding to thalidomide. And we confirmed that the double point mutant does not bind to thalidomide using our affinity-based technology. And the co-immunoprecipitation experiments show that the Cereblon forms the E3 victim ligase complex. And Western blotting show that the thalidomide is ubiquitinated. And as expected, however, its ubiquitination is resistant to thalidomide. I show here the expression of the double point mutant of Cereblon suppresses thalidomide-induced teratogenicity in zebrafish. So embryos, expressing the exogenous wild-type Cereblon was still sensitive to thalidomide, whereas the embryos exogenously expressing the double point mutant were resistant to thalidomide and developed apparently normal fins and aortic vesicles in the presence of thalidomide. So these results indicated that Cereblon is a bona fide target of thalidomide teratogenicity. So to further confirm this, we used chicken because chickens are an established animal model for thalidomide teratogenicity, and they are anatomically closer than zebrafish to humans. So these experiments were performed in collaboration with Professor Ogura and Assistant Professor Suzuki at Tohoku University. We first made sure that the chicken Cereblon can bind to thalidomide and interact with a human DDB1. And we administered this thalidomide and delivered foreign gene by electroporation into this region of the egg, which develops into the forelimbs. So administration of thalidomide strongly inhibited the formation of the forelimb or wings. So we also found that the exogenous expression of the double point mutant significantly suppresses thalidomide-induced teratogenicity in chicks. So therefore, our findings in zebrafish were validated in chickens. So here is a summary of our major findings up to this point. We identified Cereblon as a thalidomide binding proteins using our affinity-based technology, and Cereblon forms an E3 victim ligase complex and contribute to the normal development of the limbs and aortic vesicles in a zebrafish model. Thalidomide exerted teratogenic effect by binding to Cereblon and altering its E3 victim ligase activity. So this work has received great responses around the world, and they led to our collaboration with Cereblon in the United States, Cereblon Corporation in the US. So and now I would like to talk to you about the immunomodulatory drugs, IMiD for short. So IMiDs are structural and functional analogs of thalidomide, including lenalidomide and pomalidomide. And lenalidomide and pomalidomide are far more effective against multiple myeloma than thalidomide. So the question here is whether lenalidomide and pomalidomide interact with Cereblon. So to address this question, we tested the physical interaction of IMiDs with Cereblon. We roughly estimated the binding affinity of IMiD for Cereblon using the competitive drug addition assays as shown earlier. And we measure the amount of Cereblon eluted from the bound fraction on thalidomide immobilized beads by adding the indicated amount of IMiDs. So the result indicated that lenalidomide and pomalidomide bind to Cereblon with about 10 times higher affinity than thalidomide. So we analyzed the binding affinity using the fluorescence-based thermal shift assay, which is a more quantitative assay, using the fluorescence dye to monitor protein thermal unfolding. Image binding affinities can be assessed from the shift in the unfolding temperatures. So the IC50 of lenalidomide and pomalidomide is about 3 micromolar. And that of thalidomide is about 30 micromolar, so supporting the competitive drug addition assay. We then elucidated the 3D structure of the IMiDs and the human Cereblon complex. So we found that the glutaramide ring common to the IMiDs is inserted into a tryptophan pocket in the C-terminal region of Cereblon. Here, I would like to talk to you about the optical isomers of IMiDs. Lenalidomide and pomalidomide have the same optical isomers as thalidomide. So this part was performed in collaboration with Professor Norio Shibata at the Nagoya Institute of Technology and Professor Toshio Hakushima at Nara Institute of Science and Technology and Cereblon Corporation. So thalidomide has an asymmetric carbon leading to two isomers, S-thalidomide and R-thalidomide. In 1979, Braske proposed a hypothesis that S-thalidomide causes the teratogenic effect and R-thalidomide causes the sedative effect. But this had remained controversial. So if Braske's hypothesis is correct, Cereblon should be able to distinguish between S-thalidomide and R-thalidomide and bind specifically to S-thalidomide. So we investigated the interaction of Cereblon with S-thalidomide and R-thalidomide. So this is competitive drug addition assays, as described earlier, show that both enantiomer bind to thalidomide, but S-thalidomide bind to Cereblon with several times higher affinity than R-thalidomide. So this was confirmed using the D-thalidomide, which contained deuterium at the chiral center and hence is hardly racemized. So we then tested the ability of thalidomide enantiomers to inhibit Cereblon autoubiquitination using the 293 cells expressing flagged HA double tagged Cereblon. And the result indicated that as thalidomide inhibits cerebron autoubiquitination more strongly than R-thalidomide. We also tested the teratogenic activity of thalidomide enantiomas using the zebrafish embryos. The result indicated that treatment of S-thalidomide resulted in a higher incidence rate of teratogenicity than that of R-thalidomide. And we analyzed the 3D structure of the complex of cereblon with S-thalidomide and R-thalidomide. We compared the structure of free form and the cerebron binding form of the thalidomide enantiomas, which are superimposed as shown here. So the result show that both enantiomas bind to cereblon, but S-thalidomide fits more stably into the tryptophan pocket of cereblon than R-thalidomide. So this is our conclusion regarding the optical isomers of image. Biochemical and 3D structural analysis indicated that S-thalidomide has a much higher affinity for cereblon than R image. However, images are known to be easily racemized under physiological conditions. So therefore, S image will preferentially bind to cereblon, and then the remaining R image will be readily racemized, leading to a supply of S image. So we concluded that the optical isomers of image are not a critical issue for their drug actions. So the next question was whether cereblon is involved in the anti-cancer effect of image. So to address this question, we investigated the association between cereblon and the anti-cancer activity of image. We used multiple myeloma-derived OPM2 cells, which are sensitive to image, as shown here. And cereblon knockdown, which was confirmed by Western blotting, resulted in the resistance of OPM2 cells to pomalidomide. So indicating that cereblon is involved in the anti-cancer activity of image. Our research group at the Mayo Clinic also reported the same result. In the previous studies and our result indicated that IL-4, CMYC, which regulate each other reciprocally and are essential for the survival of the multiple myeloma cells, are downregulated. But the P21, which inhibit the cell progression, is upregulated when treated with image. But these image-induced gene expression changes were not observed upon the silencing of cereblon, so indicating that image regulate the expression of IL-4, CMYC, and P21 via cereblon. So we have identified the mechanisms of the main actions of image via cereblon. Two groups in Boston have also independently reported the same result. And I will not go into in detail on UV scan analysis, which is a method of identifying the ubiquitinated peptide. But we'll just give you a little, just a simple outline. And multiple myeloma cells were treated with or without image, and the ubiquitinated peptide were collected by immunoaffinity chromatography using a specific antibody and analyzed by tandem mass spectrometry. And the ubiquitinated peptide enriched by treatment with imide was screened. And we have identified IROS as a novel cereblon substrate, which shows a significantly increased ubiquitination when treated with image. So Icaros and Iros, which are members of the Icarus or IKZ family encode zinc finger transcription factors. That's important regulators of lymphoid development and differentiation. So we first investigated the effect of image on T cells. And lenalidomide and pomalidomide treatment of T cells induce the degradation of Aiolos and Icarus, as shown here by Western blotting. And this degradation was inhibited by treatment with MG132, proteasome inhibitors, or Cereblon knockdown. So these results indicated that the Cereblon-mediated ubiquitination is involved in the degradation of Aiolos and Icarus upon IMiD treatment. So we then tested the effect of image on the interaction of Cereblon with Aiolos and Icarus. And the flag-tagged Cereblon was co-expressed with Aiolos or Icarus in the multiple myeloma-derived KMS12 cells or 293 cells. And the cells were treated with or without image, and these cell lysates were prepared. So treatment of KMS12 cells with pomalidomide, lenalidomide, and thalidomide similarly induced the interaction of Cereblon with Aiolos. And when the flag-tagged Cereblon was immunoprecipitated using anti-flag antibodies, and Aiolos and Icarus were co-immunoprecipitated with wild-type Cereblon, but not with a double point mutant. Only the cell had been treated with the pomalidomide. And the same result were obtained for lenalidomide, although I didn't include the data here. So these results indicated that the binding of image to Cereblon recruits Aiolos or Icarus to the Cereblon E3 ubiquitin ligase. And Aiolos and Icarus are known to be repressors of IL-2. So here I show the silencing of Aiolos or Icarus enhances IL-2 expression, which mimics the effect of treatment with image. The combination of silencing, combination of Aiolos or Icarus silencing and treatment with image further enhanced IL-2 expression. So these results indicated that the Cereblon-mediated degradation of Aiolos and Icarus is responsible for the immunomodulatory effect of image via enhanced IL-2 expression. We then investigated the effect of image on multiple myeloma cells. And Aiolos and Icarus are known to be activators of IL-4, which is a protein essential for the survival of multiple myeloma cells, as I showed earlier. So here I show the silencing of Aiolos and Icarus lower the cell viability of multiple myeloma cells, which mimics the effect of treatment with image. So Cereblon-mediated degradation of Aiolos and Icarus is responsible for the anti-cancer effect of image. So I have illustrated the mechanism of action of image. So image induce the ubiquitination and degradation of the Aiolos and Icarus via Cereblon E3 ubiquitin ligase, leading to T cell activation via enhanced IL-2 expression and the loss of multiple myeloma cell viability via reduced IL-4 c-MYC expression. So more specifically, just two types of substrate have been identified. And the binding of image to Cereblon alters its substrate specificity, leading to both the inhibition of ubiquitination of its natural substrate, including slow, MICE2, and Cereblon itself, leading to their accumulation, and the induction of the ubiquitination of novel substrates such as Aeros and Icaros, leading to their degradation. And an ubiquitination of slow, which is normally ubiquitinated and retained in the ER, is transported to the plasma membrane, leading to enhanced potassium channel activity, which might be involved in epilepsy. And accumulation of MICE2 by a similar mechanism might cause the limb hypoplasia. So recently, a novel lenalidomide-specific substrate of Cereblon has been identified. Aeros and Icaros are substrate of Cereblon common to thalidomide, lenalidomide, and pomalidomide, which are used for the treatment of multiple myeloma. And on the other hand, the enzyme casein kinase 1A1 or CK1 alpha has been identified as a novel substrate of Cereblon, which is a highly effective treatment for myelodysplastic syndrome called MDS for short, with deletion of the long arm of the chromosome 5 or 5q. So the CK1 alpha is known to phosphorylate and inactivated p53. So lenalidomide specific degradation of CK1 alpha activated p53 and induces apoptosis in the cells. So the CK1 alpha gene is located in the region of 5q that is deleted in the patient with MDS 5q minus. So this haploinsufficiency might explain the therapeutic efficiency of lenalidomide for these diseases. So more recently, the third generation derivatives of thalidomide have been developed. One of the derivatives, CC122, which was found to exert a favorable therapeutic effect against diffuse large B-cell lymphoma via Cereblon, but its substrate is not yet known. So I would like to propose a mechanistic model of the multiple pharmacological effect of thalidomide and its derivatives. So we initially thought that the multiple pharmacological effect of thalidomide and its derivatives are a result of their binding to a different target, as shown here. But our recent findings have provided the data against this old model. So we now propose the current model in which the thalidomide and its derivatives bind to a single target, Cereblon, and that the alteration of the substrate specificity of Cereblon induced by the binding of thalidomide and its derivatives is thought to be essential for the multiple pharmacological effect. Okay, so and last but not least, I would like to thank the following colleagues and collaborators who participated in this research. And thank you very much for your kind attention. Please.